We're moving on to a talk that I've particularly been looking forward to by Dr. Matt Evans, who's a consultant pathologist in Southampton and runs the Central and South NHS Genomic Laboratory Hub, and is going to be talking to us more about molecular profiling of biliary cancers.
Well, thanks very much for that introduction, and thank you as well for the invitation to speak. It's an enormous pleasure. What I'm going to be doing here is building on what John has already spoken about in terms of those targeted treatments and immunotherapies by focusing in more on the testing which allows those treatments to be given. So I'm going to be talking about molecular profiling of biliary cancers. And I think it's worth just spending a moment before I start talking about this in detail, just saying that if I'd given this talk 5 years ago, it would have been over in about 2 or 3 slides. There really was very, very little to talk about in this area. Fast forward to today, and all of a sudden we have large numbers of biomarkers with many, many more on the horizon. And I think in that respect, biliary cancers are pretty unique. I don't think we've seen another cancer, at least that I can think of, where we've seen such an enormous proliferation of testing in such a short period of time. And obviously, that implies lots more treatment options. So what I'm going to do is try to address 3 key questions. So I'm going to start by asking, what types of molecular alteration do we actually look for? What do they look like? What do they mean? From that, I'm then going to talk a little bit about the technologies which we can use to identify those alterations. So what are the technologies and the different types of tests? What are the strengths, weaknesses? Why might you pick one over another? Because there are lots of options out there. And then in the last couple of minutes, I'll say a few words about what we do with the results. Now, obviously, that is more on the side of the oncologist, but I'm going to make the argument that actually that those results are very important diagnostically for pathologists as well. So, starting from the top, let's start by thinking about the different types of biomarker which we test for. And this is in general for all solid cancers. In the majority of cases, when we're talking about a genomic alteration, so an alteration at the level of DNA or RNA, they mostly come in 3 types. So at the one end of the spectrum, you have the small sequence variants. These are little spelling mistakes in your genome, so little spelling mistakes. So you can see there, we've swapped one letter for another, we've added in a few letters, we've taken out a few letters, but they're all pretty small. And in real life, that's the kind of thing that you will be looking at. So you can see that we should have a T in our DNA sequence, but we've substituted it for an A, we've removed a few letters, we've added in a few letters. So small sequence variants. And these are the kinds of things which we're looking for in genes like, for example, IDH1, BRAF, KRAS. Then you come on to the structural variants. Now, these are entirely different. We're not talking about little spelling mistakes here. What we're talking about here is where you take a big chunk of text from your genome and move it to a completely different location. So you can see there, I flipped our 2 lines around. This is more than a little spelling mistake. And in real life, it might look something like this. So you've got a chunk of DNA on one chromosome. If you were to move that chunk of DNA to a different chromosome, that would be a structural variant, otherwise known as a fusion, rearrangement, translocation. They all mean similar kinds of things. And these are the kinds of things which we're looking for when we talk about genes like FGFR2, NTRK1, 2, 3. And then finally, we have the copy number variants, which are kind of what they sound like. It's where you have an abnormal number of copies of a particular region of the genome. Usually, it's the case of having excessive numbers of copies. So you can see there, we've got 2 extra copies of one line of our text, and in real life, it would look something like this. You have a chromosome with a region on it. You should normally only have 1 copy of that region, but you can see here we've got an amplification where we've ended up with 4 copies of it. And that's the kind of thing that we're talking about when we talk about things like HER2, although HER2 can get a little bit complicated. Now, that's made it look very straightforward. You know it's not that simple. There are actually lots and lots of other types of alteration which we test for, not necessarily in routine practice in biliary cancer yet, but there are lots of alterations out there which we look for in solid cancers. And I would just like to take a moment to stress that that one at the end, which I've helpfully put in grey, the protein expression abnormalities, these are the non-genomic alterations. And I think it's fair to say that these have been a little bit neglected historically. They don't seem quite as exciting as the things that you look for in these big sequences. But I have to make the point that that's where we really are seeing a huge amount of growth in biomarkers across all cancer types. And so I think that's an area where we do need to focus. So I've laid out what the main types of alteration are, particularly the small sequence variants, the structural variants, the copy number variants, plus all the others. How do we go about testing for them? Well, let's start from the top, and let's start by thinking specifically about the alterations which we need to know about in cholangiocarcinoma. One of the things which we need to know about in a cholangiocarcinoma to get patients on the right treatment and to establish their prognosis is the status of the mismatch repair or MMR system, also kind of confusingly interchangeably described as mismatch repair as microsatellite instability or MSI testing. The idea here is that whenever you replicate DNA, you will inevitably get errors. That's normal. But we have a very helpful little system in our cells called the mismatch repair system, which is made up of a number of proteins which will come along, they will identify those mismatches, and they will correct them. So they'll turn it to the correct base, meaning that if your MMR system is working normally, then your DNA will replicate accurately. If, however, there's a problem with that system, so there's a problem with any of the components of that system, suddenly you won't be able to repair those errors. They'll accumulate and accumulate and accumulate, and you could see how that could result in a cancer, including cholangiocarcinoma. The reason that this is important, going back to what John was talking about, is that knowing whether this system is working normally or not is important in working out how likely a patient is to respond to immunotherapy. Now, confusingly, there are two different ways of testing to see whether that system is working normally. On the one hand, you've got MMR immunohistochemistry, which is what somebody like me does in my lab, and I look down a microscope to give a diagnosis. Or you can extract DNA from the tissue and undertake microsatellite instability testing. The terms are used interchangeably. They're two different ways of looking at the same thing. And just to give you a flavor of how these things work, when we talk about MMR immunohistochemistry, what our scientists will do is that they will cut very, very thin slices of the tumor tissue. They'll put it on a glass slide, and they will stain each of those slides with a different MMR protein. Now, I can pretend that this is very, very complicated, and in some cases it is, but in most cases it boils down to this. If all 4 of your MMR slides look brown, it's MMR proficient, so your MMR system is working normally. If any of them look a bit blue, like those ones, in general, that means your MMR system is not working normally. Therefore, there's an MMR defect, there's a problem with the MMR system, the patient is more likely to respond to immune checkpoint inhibitors. I'd like to pretend that it's more complicated than blue and brown, but often that's as complicated as it gets. The other way that you can test for this is by microsatellite instability or MSI testing. And I'm not going to go into this in a lot of detail, but basically you plot lots of curves based on the lengths of structures called microsatellites in your genome. If they all look like nice smooth curves, that's telling you that your tissue is showing microsatellite stability, so your MMR system is working normally. If you see that they have double peaks, then that's telling you that there's microsatellite instability, that your MMR system is not working normally. Two different ways of looking at the same thing. And just to say, and I'll come back to this a little later on, you can now also assess microsatellite instability status using next-generation sequencing, which I'll come back to later on. Now, as to which of these 3 approaches is the best, the jury at the moment is out. In my opinion, we don't have enough evidence, and we need more research on this area to work out which is the most accurate way of assessing this thing. So that's the first of these markers which we need to assess, MMR, MSI. But as you know, that's not the extent of it. We've seen a large proliferation in biomarkers in cholangiocarcinoma in recent years, and we now have a host of other alterations which we need to assess for in routine practice. IDH1, small sequence variants, little spelling mistakes, FGFR2, and to a lesser extent, NTRK rearrangements. Now, to make things a little confusing, for each of these different alterations, there are different approaches which you can take to your testing. On the one hand, you can do what we've historically done, which is to use one test for each gene of interest. So you want to know about IDH1, do an IDH1 test. You want to know about FGFR2, do an FGFR2 test. Those technologies have served us very well, but increasingly we're moving over to a next-generation sequencing approach. This is where you use a single next-generation sequencing test which will look at multiple different genes in parallel, and it will actually look at multiple samples all at the same time, making it a very efficient way of testing large numbers of samples. And as I say, increasingly we're moving from this single gene approach to the next-generation sequencing or NGS panel approach. What I'm going to do now is just talk a little bit about a few examples of single gene approaches which you can use for testing, and talk a little bit about how next generation sequencing works. So, let's start by thinking about a couple of the single gene technologies which you can use to pick up alterations in cholangiocarcinoma. One of the most commonly used technologies, single gene technologies, which you can use to assess for small sequence variants, like those in IDH1, is real-time PCR. And it's quite a straightforward technique in many ways. Let's imagine that you've got a strand of DNA and it's got a mutation in it. So that's our T there, which I've highlighted. You will then apply to your extracted DNA multiple different probes, each with a different color attached to them, which will all bind to different mutations which are of interest in your sample. If you apply those probes, you'll see that one of them will bind to that sequence, that will release that color. You'll look at that using a detector, and if you see that color, that's telling you that that particular mutation is present. It's a really robust technique, makes a lot of sense. The problem is that you can't have an infinite number of these probes in your test, meaning that rare, weird, and wonderful things you will end up missing. FISH, fluorescence in situ hybridization, is another targeted or single gene technology which you can use to pick up structural variants. And the idea here is that you take a cell, you apply different colored probes to that cell, one of them which binds to the gene of interest, say for example FGFR2, the other binds to a region of the genome immediately next to your gene of interest, meaning that under normal circumstances, those two probes will be next to each other. So you can see there you've got your green and your orange sat next to each other. If, however, your FGFR2 gene is rearranged, so it moves to a different location in the genome, you'll see that two of those colors will be split. That's telling you there is an FGFR2 rearrangement present. Makes loads of sense, sounds very, very straightforward, and often it is a very good technology. Problem is it can give you false negatives, it can give you false positives. It's not entirely foolproof. And it's partly because of those downsides that we're increasingly, as I say, moving over to next generation sequencing. And the way that you will normally profile a solid cancer is that you'll run a DNA NGS panel to look for small sequence variants. So you would do that to look at your IDH1 plus BRAF plus KRAS plus anything else you might be interested in, because if they're on the panel, you can look at them at the same time. And then you'll often use an RNA panel at the same time to look for your structural variants, so your FGFR2. So rather than looking at multiple different genes using multiple different tests, you're using two tests to look at the same number of genes and potentially even more. Now, I mentioned next generation sequencing panels because a next generation sequencing test will look at a particular set of genes all at the same time. Those panels can be small, like that, where you've got maybe a few dozen genes. They can be very large, so where you're looking at 500 genes or so, and they can potentially be whole genome if you really need them to be. Clearly, going for large panels is going to give you more information, and it's going to be particularly helpful for getting patients onto clinical trials. And just to mention, with large NGS panels, you can also assess for things like MSI, so it's kind of a one-stop shop if you can do it. So, which is the best method for profiling a cholangiocarcinoma? It kind of sounds like I've implied that it's going to be a large NGS panel. And that's kind of true to an extent. But I would say that all of these different approaches have different strengths and weaknesses. And so the best method will depend on the clinical situation. If you have a very, very poorly patient and you just need a result as quickly as possible, it may be that you'll need to go for one of those old-fashioned single gene technologies. If that identifies something, job done. If it doesn't, then you might need to do some more testing. If you've got a patient where you're considering a clinical trial, well, you need to use a large NGS panel because you need that rich information. So, in the last couple of minutes, what do we do with the results which we obtain from these tests? Well, that's an example of one of the reports from our genomic laboratory. Slightly small print, but you'll see there that you have the result under report summary telling you that an FGFR2 gene fusion has been detected. You have then lots of description of what that molecular alteration is, what it means. Then you have some details about how the test was done. And on the last page, you have a list of all of the different genes which were covered using that test, so you know where there aren't alterations. Now, I'm not going to tread on the oncologist's toes by talking about what you should be prescribing off the back of these alterations, but clearly these things are very important from a predictive point of view in terms of making sure that patients end up on the right treatment. But as I said before, I don't think this is just about getting patients on the right treatment. I think it's also about getting patients an accurate diagnosis. And I'm sure my colleague Tim Kendall up in Edinburgh will be telling us more about the challenges of making a diagnosis of cholangiocarcinoma later on. The point I want to make here is that when I showed you this report a moment ago, I was a little bit dishonest because actually I sent this to my genomic laboratory thinking that it was a lung cancer. You can see there, that was my diagnosis, lung adenocarcinoma. It turned out that there was an FGFR2 gene rearrangement. That's very, very rare in lung cancer. And so we actually changed the diagnosis in this case to cholangiocarcinoma. Patient ended up on targeted treatment, and I'm pleased to say is doing well today. So this testing also has importance in getting the diagnosis right. So I think this all comes down to a question of what is a diagnosis. There was a time when just making a diagnosis of cancer was seen as acceptable, cancer of the liver, adenocarcinoma in the liver, and I think today we've been at the point where making a diagnosis of cholangiocarcinoma is seen as acceptable. I think we need to be moving to a position where this is seen as the standard type of diagnosis which we give, and if we're giving less than that, we're not giving a full diagnosis. But clearly, that's going to require a lot more testing. Thank you very much.